Our next speaker is Chelsea Hess, and she's from North Carolina, where she received her BS in marine biology from UNC Wilmington. While she was there, she worked as the sea turtle monitoring coordinator and aided in the marsh monitoring program for the North Carolina Coastal Reserve and National Estuarine Research Reserve. Since 2015, she has attended Louisiana State University and is currently a Ph.D. candidate in the lab of Dr. Fernandez Galvez, excuse me. And her dissertation research focuses on the multi-generational physiology and morphological effects of crude oil exposure in fish. So without further ado, Chelsea. Either advance through just that way or advance through. Okay, but the pointer just.
Yeah, that's fine. Thank you for the introduction. Again, my name is Chelsea Hess. Some of you may be confused. I am actually stepping in for Dr. Ben Devansky, who could not be here. However, he did get his PhD from the lab that I'm currently working in. And a majority of today's presentation covers his work while in the lab, which has essentially been the foundation of my dissertation research. And today, like we said, I'll be talking about the impacts of crude oil on the physiology of estuarine fish in the case of the Deepwater Horizon oil spill. And I'm not going to inundate you with a ton of DWH facts, many of which we already know in this room, but I do want to point out that as a result of the Deepwater Horizon oil spill, 1,770 kilometers of coast in the northern Gulf of Mexico was oiled as a result. Not only that, but 790 of those kilometers were oiled directly in my home state of Louisiana. And this event spurred many scientists to look at the effects of crude oil on marine and estuarine organisms, including the Galvez lab and our collaborators who have been looking at the effects of crude oil on fish physiology since this event. And to address the effects on fish, we use the model organism, the Gulf killifish, Fundulus grandis. And if you've ever been fishing in the northern Gulf of Mexico, you've probably used these as your bait fish. They're the most abundant vertebrates in the coastal marshes of the Gulf of Mexico. And due to their relatively small size, they're easily reared in a laboratory setting, which is great for us. Additionally, they produce transparent embryos, which allows for easy monitoring of development. And the Gulf killifish, along with their sister species, the Atlantic killifish, have long been used for looking at the effects of environmental stressors. Not only this, but they're an ecologically relevant model because they're one of the many organisms that were impacted by the Deepwater Horizon oil spill. And here we have an image taken by Ben Devansky in Grand Terre, Louisiana, following the spill, showing the interaction of adult Gulf killifish with the oil. And unfortunately for this fish, along with many other species, peak oiling coincided with the peak spawning event, resulting in oil exposure to not only adults, but also juveniles, larvae, and embryos. So given this situation, the Galvez lab and collaborators set out to answer the following experimental questions. First, what are the physiological and molecular impacts of oil exposure at different developmental stages in fish? Second, to identify the geographical and temporal span of oiling in the field. And finally, to see how oil exposure history affected sensitivity of fish to the DWH oil. And to address this first, fish were sampled in field collection sites, 6 different sites along the northern Gulf of Mexico, including Grand Terre, Louisiana, Bay St. Louis, and Bellefontaine Point in Mississippi, and then Bayou La Batterie, Fort Morgan, and Upper Mobile Bay, all indicated on this map here. And they were sampled at 3 different time points following the oil spill, including once prior to oil reaching, once again in June following oiling, and as indicated on this figure here, black is up to 4 kilometers distance from surface oil, where red indicates 0 kilometers distance from surface oil. And then finally in September of 2010. I do want to note that Grand Terre was the only location that oil made landfall, and this was between June 9th and 14th of 2010. And sediment sample analysis showed that there were extremely high concentrations of oil constituents like polycyclic aromatic hydrocarbons, or PAHs, which are extremely toxic and monitored by the EPA. And the approach was to measure tissue-specific changes in gene expression associated with oil exposure. And to do this, one of the approaches used is what's called a microarray. And what this does is it's an approach that allows us to measure the relative increase or decrease in gene expression following some sort of treatment or exposure. And what happens is mRNA, or messenger RNA, which is basically just the recipe book provided by the DNA to inform protein synthesis. That is then labeled with fluorescent tags and then placed into microscopic wells on a chip that have complementary strands of mRNA in them. So we can get an idea of gene expression based on difference in colors based on the fluorescence tags. And this is a lot, but what I do want to point out is that the differences in colors identify differences in gene expression, where this bright yellow is a relative increase in gene expression, and the blue is indicative of a relative decrease in gene expression. Here we have the 6 different locations in which fish were collected. And for each location, there are 3 columns right there. And that's indicative of each of the 3 sampling points. And first thing they had to do with this study is identify, okay, well, did oil exposure take place? And can you measure that on a genomic level? Well, it turns out, yes, it did. And what we see here is in the Grand Terre, Louisiana population, the only population where oil made landfall, we see this strong increase and decrease in a set of genes associated with what's called the Aryl-hydrocarbon receptor pathway. This seems like a lot, but I promise we will get through this together. But this differential regulation that's not seen at the other sites coincides with the time and location of oiling. And the Aryl-hydrocarbon receptor pathway is really, really important for breaking down drugs, outside compounds, and including things like oil. So here we have an example of one of these polycyclic aromatic hydrocarbons, or PAHs. And when that oil constituent interacts with what's called the Aryl-hydrocarbon receptor, which sits on the outside of the cell, it is then chaperoned to the nucleus, where it binds with something conveniently called the Aryl-hydrocarbon receptor nuclear transporter. Long name, but sometimes scientists make logical names for things, which is great. And that dimer then binds to a portion of the genome called the drug response element. In binding, it alters this messenger RNA, thus altering our gene expression. So from this microarray, we saw differences in regulations of these genes associated with this pathway, which I would like to add is present in you and me as well, and most other vertebrate species. So this isn't unique to fish. But in particular, I want to point out this gene right there called cytochrome P451A. That's a mouthful, but it's really important, and it's one of the major metabolizing steps for the PAHs. So Ben Debansky took it another step further and looked at the presence of the CYP1A protein in gill epithelium from Fundulus grandis in Grand Terre, Louisiana. And what he saw was that at trip 1, prior to oiling, there's limited kind of these red cells, which are indicative of that protein being expressed. However, in trip 2 and 3, in June and September, they see a lot of this red dye indicating an increase in that CYP1A, meaning there was oil exposure and it is responding to that. What's most interesting is that nearly a year later, there's still a high induction of this CYP1A, indicating continued stress even a year out. So the identification of exposure to toxic components in oil was achieved. Now we wanted to look at what physiological effects could there be implicated from these changes in gene expression. And first, they looked at reproduction. And again, you don't need to know the name of these genes. However, it's very clear if you look at the Grand Terre population that genes important for both egg and yolk development were essentially turned off, coinciding with the timing and location of oiling. Additionally, if we look at another pathway of genes, we see that activation of the AhR pathway results in increased ubiquitin ligase activity, which is an enzyme that's really important for the degradation of sex hormone receptors. So increased degradation of these receptors results in suppression of sex hormone-induced gene expression. So based on this microarray data, they then said that, yes, there is potential for compromised reproduction as altering the estrogen cycle. And finally, they looked at impacts on cardiovascular system. And what they found, again, this Grand Terre, Louisiana population showed differential regulations of genes associated with cardiovascular development and function, resulting in altered cardiac physiology. So in conclusion, altered gene expression resulting from oil exposure occurred in the Grand Terre population, and this gene expression data suggests potential for altered reproductive capacity and alterations in cardiovascular fitness. And following this, the Galvez lab, in part with Dr. Ben Debansky, went a step further to identify the developmental impacts of the crude oil exposure on killifish. And to do this, Ben, having a great time in the heat in the northern Gulf of Mexico, took sediment samples from 3 locations. He took samples from Grand Terre, Louisiana, once in 2010, and again in 2011. The more northern place right here in Wilkinson Bay, which had lesser oiling. And then there was a reference site, Bay Saint Bois, that was kind of used as the control. And if you can take a look at the right-hand side of the screen, on the y-axis, we have total PAH concentration, or polycyclic aromatic hydrocarbon concentration. And that increases as we move right across the different sites. Note that the right 2 sites are Grand Terre in 2011 and 2010. And then Gulf killifish embryos were then subjected to either saltwater with this clean reference sediment or saltwater and oiled sediment. And interestingly enough, the heavily oiled sediment resulted in lower relative heart rates. If we look at heart rate on the y-axis, and then the sites in order of increasing oil concentration on the bottom axis, we see that the Grand Terre sediment that was exposed to the embryos resulted in significantly decreased heart rates. Not only that, but this heavily oiled sediment resulted in decrease in hatching success in Gulf killifish embryos. We have percent of total on the y-axis, where the green indicates the percent of that total that actually hatched, which is significantly smaller than in the other 3 locations. And additionally, the heavily oiled sediment resulted in delayed time to hatch or increased time to hatch, where the red and black or Grand Terre, Louisiana sediment exposed embryos took longer to hatch, which can have huge implications for fitness and survivalship. And finally, Dr. Ben Debansky identified developmental abnormalities, where this is a normal heart, and the other 2 show elongated hearts or improper folding, the presence of pericardial edema or swelling around the heart, craniofacial abnormalities, and hemorrhaging. So in conclusion, we found that exposure to oiled sediments result in increases in time to hatch, as well as decrease in hatching success, and an increase in incidences of teratogenic effects, or these developmental abnormalities, along with impairments to cardiovascular fitness. And these results were substantiated by another paper by Incardona et al., which saw that in zebrafish, a freshwater species commonly used as a model for human health, that PAH exposure resulted in subtle changes in the heart, but it had huge implications and resulted in significant decrease in swimming abilities even a year after embryonic exposure, suggesting that it's due to decreased cardiovascular abilities. And my dissertation research focuses on not only identifying the long-term effects of crude oil exposure, but the effects that span multiple generations. And with the Gulf states, great stake in both fisheries and oil exposure, it's really important to understand what oil exposure means to not only the individual, but the children and potential grandchildren of that individual, whether it's fish or human. And since experiments on humans are often frowned upon, I still work with the Gulf killifish. And if we look at the top here, we have a normal Gulf killifish. This is a male. However, if you look right below it, this is a killifish that had parents that were exposed to oil. So it only had direct oil exposure as egg or sperm. And what we're finding is that oil exposure, particularly when the father's exposed to oil, there's an increased incidence of both jaw deformities and spinal deformities, which impair both live prey capture and swimming abilities. And you can see this scoliosis apparent in CT scans at the bottom, showing that there's gross developmental differences following parental oil exposure. What's interesting, though, is that with parental oil exposure versus the embryonic oil exposure, as discussed in the earlier study, there are no significant differences in hatch or heart rate. So if this exposure stopped at embryonic development, then we may have concluded falsely that there was little to no toxic effect. So this kind of calls for the need for more sensitive early biomarkers of toxicity. So in summary, we showed that oil exposure following the Deepwater Horizon oil spill resulted in differential gene regulation that are implicated in reproduction and cardiovascular fitness. That embryonic exposure to oil-rich sediments can result in decreased hatching, increased time to hatch, and increased incidence of developmental abnormalities. And that it's a huge importance to identify the long-term multi-generational effects of crude oil exposure as we move forward and respond to future spills. And I would like to acknowledge my boss, Dr. Fernando Galvez, and former and current lab mates, Ben Debansky, Charles Brown, and Tiffany Sims, and our collaborator, Dr. Andrew Whitehead, in his lab at UC Davis, along with all of our funding. Thank you.